everyone. Welcome back to this edition of Meet iToBoS, where you get to meet in a little bit more depth single consortium partners. iToBoS stands for Intelligence Total Body Scanner, and the Melanoma Patient Network Europe is one of the project partners. Feedback we have regularly gotten from our community was that people were very excited about what's going on in the project and wanted to know more. And we are now trying to find a good way to give everyone the opportunity to have access to the consortium partners, to learn more about who they are, what they do, and to ask questions. So you're very much invited to participate in this session. So this is a particularly interesting project because it is so broad. So it has highly technical engineering parts, it has fancy lenses, and that's the topic we'll be talking today about with Xavier. Then we have artificial intelligence, we have ethical concerns, we have clinical trials. So it is really broad and exciting to see what one can build together if one brings different disciplines together. So with that, I would like to hand over to you, Xavier, if you could tell us a little bit about who you are, your background, how you ended up in iToBoS, and what you're now doing there. Over to you.
So hello, everybody. My name, as you well said, Xavier. I come from Spain, and originally I studied engineering, in particular telecommunication engineering. And then in my master's, I went already towards the optical field with a master's in photonics, and also I did a PhD in photonics. But that was really deep physics. And then when I finished the PhD, I made one step back into real life and entered Optotune. Optotune is an optics company, as I will explain a bit more during the talk today. And in the project iToBoS, as you will see, we are the ones building the lenses. So one of the big points of the project is to be able to have high resolution images of the patient lesions, and that's where we enter. I think it's the moment to share my screen, and we can go towards the presentation. Everybody sees the screen now? Okay. Then I'll start. So as I was saying, today I'm going to talk about the optical challenge of iToBoS and how we can solve it with what we call liquid lenses, which is the star product of Optotune. The agenda, let's see if I can change slide. Yeah. The agenda for today is the following. So first, I will start with the general objectives of iToBoS, and from there, I will go deeper and deeper until we find what is the optical problem that we face in the project. Then I will jump into Optotune's technology and why this is the solution that we think is the best for this optical challenge. And finally, the lenses are a product that we have off the shelf, but that was not enough. We still needed to do some work on them to be able to overcome all the challenges. And I will talk about the two tasks that Optotune as a partner of iToBoS had in the project. So let's start with the general idea on the iToBoS project. As you know, the iToBoS project has as a broad objective to generate a tool that is able to give a better diagnostics or better help to diagnostics to the physicians on when a mole is melanoma or it's not. This has different facets to it. The first one is developing a hardware. So we want to build a new full body scanner. Until now, you know that we have handheld dermatoscopes that can take very high resolution images, but those are local images that need to be labeled and located in the patient's body. And then we have also some full body scanners that have low resolution. And the two of them can be used together, and they give us very nice tools. But what we want to do now is to have the high resolution and the full body avatar all in one. That's the first job that we have in the iToBoS project. The second one is to help the clinicians to analyze all that data. I mean, until now, clinicians have a lot of experience. They have gone through many cases, but we want to use artificial intelligence and generate what we call a cognitive assistance that will mix this dermoscopic data that we take through the pictures, but also will mix genomic data from the patients, their medical records, family records, even data from previous visits, all together to give to the clinician the best help possible. Also related to the artificial intelligence, there is also another point. Usually artificial intelligence systems have what is called the black box effect. So basically, they give an answer, a probability, or a black and white answer, but there is not much about why that answer has been calculated. And there is a new field in artificial intelligence that goes into that, trying to answer this question. And instead of giving just this probability answer, they try to give also some explanation of what part of the data has been important to take that decision. This is what on the technical side of the solution, but then of course, this has to be really tested in real life. For that, the project will also acquire a big data set of skin lesions of different patients so that we can train these systems. You know, artificial intelligence systems always need a lot of data to be able to make good predictions in the future. And finally, of course, we cannot avoid this. This is technology that has to go in real life in hospitals. We have 3 partners, Barcelona, Trieste, and Brisbane, and they will conduct clinical trials such that we really validate that this tool is doing what we want it to do. But today, as we already said, we are going to talk about hardware. So let's dive deep into the scanner and what is the idea for this piece of equipment. First of all, the general idea of the scanner will be similar to other scanners that we have in the medical field, like MRI. So the patient will be lying on a bed and the scanner will be around. The scanner in the initial designs, which are changing, but more or less the idea is the same, will be formed by 5 arches, as you can see here, that will be above the patient. Each one of these arches will have 3 sections, as you can see here. And in each section, we have what we call a camera module. These camera modules will be able to move laterally so that they can point to different parts of the patient, and also vertically so that we can focus to the different distances on the patient. Of course, there are patients of different sizes, and also our body is irregular, so we need to be able to approach the camera more or less to different parts to take the good quality images that we need. Finally, even deeper, each one of these camera modules that I show here will have several elements. Of course, the camera, a high resolution camera, that's basic, but then also an objective lens. As you can see in this model, the camera is at a certain distance from the patient, so we need to focus to the distance. We cannot take just the camera as it is. Then we'll also have some lighting system, because we want to be able to take these images quite fast. So we want to reduce the exposure as much as possible. So we need good lighting to make sure that the cameras get still enough light. And finally, we have also distance sensors such that we can know at every moment what is the distance to the skin of the patient, and then regulate this distance that I was talking about before. Now, today, as I said, we are going to talk about optics, so we can go even one level deeper and start talking about the lenses. And before anything, I want to analyze what are the boundary conditions when we were starting to design these lenses, so that we understand how we arrive to the solutions that we have. The first of the conditions that it's clear in any medical device is the safety of the patient. There is no doubt about that. And in this case, as I said, we have some moving cameras, and they are around the patient moving at a high speed. So the first clear condition is that these cameras cannot be too close to the patient. And going through regulation, I'm not going to go there because it's not my expertise, but the solution, or the final value that we got, is around half a meter from the patient. If we want high resolution, you can already see that high distance is not the best of the conditions. So you will see some of them will be fighting against each other. The next condition is the comfort of the patient. I mentioned before MRI, and I'm sure if you have had an MRI before, it's not the best memory of all, because it's a huge machine. The patient is inside that tunnel with a lot of noise, and they last for a long time. So our goal for this project is that the scanning time is reduced to a minimum. That means that we have to take a lot of pictures, but we will try to make it as fast as possible. If we go more to the technical side, of course, as I mentioned from the beginning, we want high resolution so that we can have the best of the two worlds from the manual dermatoscopes and the big scanners. Unfortunately, when you have an image with high resolution, because we are working with images taken by cameras that have a finite number of pixels, if you want a lot of resolution, you are imaging a very small portion of the object. So it's what we call the field of view. And in this case, it's very small. And the consequence of that is that for a scanning that covers the full body of the patient, we need to take a lot of images. And you already see that here we have two competing conditions. We want to minimize the time, but we will have to take a lot of pictures. So here you see that there is already a challenge. And the final condition is the fact that our body, the human body, is a 3D object. And this is important for taking pictures. We all have our experience when we take pictures with our cameras that it's difficult to have all the objects that are in different positions with respect to the camera, to have all of them well resolved. That's related to what we call depth of field. And in our case, our situation is that we need a big depth of field. We will have the skin of the patient that is irregular, the body is irregular, some elements at a given distance of the camera, but then there are some that are closer and some that are farther, and we want all of them well resolved. Because of the large distance and the high resolution, we already know that the depth of field of our camera will be very small. So already with a single image, we cannot take high resolution pictures of the whole contrast of the body. So what we'll do is we use a technique, it's called image stacking. And this technique consists in the following. We take many pictures of the same region, each one of them focused at a different distance. That means that the first picture will have good resolution in this area, but not that good in that one. And the last picture will have good resolution here, but not that good in there. Then with some software, you can combine all those images and get a single picture where all the regions are in high resolution. That's a very nice technique, but again, it again multiplies the number of pictures we have to take by number. So we have a lot of pictures to cover the whole body and a lot of pictures to be able to have all of them resolved. So again, we are competing against having the smallest scanning time possible. With these constraints set, let's see how we can solve them. The first one, it's a classical solution. Here, there is nothing too new. We have to design a custom objective lens. It's not the trickiest of the problems, but we'll have some constraints there to be able to have good quality at the distance as we want. The second one already related to this limitation of how big the signal, the images we can take are. We are going to choose a sensor, a camera sensor that is quite big. Of course, at the same time, we want this to be a standard camera that is industrial grade because this machine should be produced in numbers in the future. So in the end, we settle for a 1-inch sensor. These are not the biggest sensor in the market, but as I said, in industrial grade cameras, this is already quite a big one. And finally, to be able to take all those pictures in the minimal amount of time, what we want is to have as fast as possible movement of the cameras, so fast scanning. That can be solved with stepper motors, again, a standard solution. But what is a bit more complex is doing fast focusing. So as I said, we are moving laterally the cameras, but also we are wanting to take a lot of pictures at different focal points. And typical lenses have big mechanical elements. So each one of the glass element has to be moved with respect to each other, and they are heavy, they are chunky. So yes, they can be moved fast, but not as fast as we would like. And here is where OptoTune lenses enter. Our liquid lenses, as I will explain in the next minutes, are able to move very fast, to change the focus very fast. And when I say fast, I mean in the range of tens of milliseconds. We can change the focal power of the lenses. And this will be key for this project. So let's dive into OptoTune's capabilities and what is the liquid lens technology. In general, OptoTune as a company, we are an optics company, that's clear, but we are not a classical glass optics company. We don't build big glass components. We take the standard ideas in optics, which are very basic, lenses, mirrors, prisms, these standard components, and build devices that make possible to tune some of the properties of these optical components. To tune those parameters, our second field of expertise is actuators. In most of our products, we have what is called voice call actuators or electromagnets. But in some of the cases, we also use more special types of motors like reluctant force motors or electroactive polymers. And even in the last years, we are introducing shape memory alloys. So putting together our knowledge of optics and actuators, we can make all kinds of tunable devices that improve the capability of standard optical components. Now, if we focus on the topic of changing a focus of lens, the solution that engineering has found until now, it's what I was telling before, these big chunky objectives that are an amazing solution, but have some limitations. And the most important one is the size and the weight of the components that make it very complex to move them precisely and fast when we need to change the properties of that objective. Instead, nature's solution is much more elegant. It's a sphere of flexible material filled with liquid and surrounded by muscles that can deform the shape of this sphere such that the rays can be focused no matter on what distance they come from. This elegant solution has not been copied until now, and Optotune did that. So we took this idea of a flexible container full of glass, of liquid, and we made it as a device, and what we call the liquid lenses. As you can already see, instead of moving big chunks of glass, we just move some liquid inside the lens, so it's much more compact. It's also much faster because the mass that we're moving is much smaller. It's reliable in the sense that we don't have tens of mechanical parts moving against each other, which have to go through wear and tear, and at the end, after some time, can break. In our case, it's just a flexible membrane that bends within the elastic limits and hence can go through thousands and thousands of cycles. We also have less tolerances. Less components means that the design is much easier. And finally, because they are driven electrically, that also gives a lot of flexibility on how we can control them. Now, if we look a bit more into the detail, it's clear that the main elements of these lenses are the liquid and the membrane. So basically, we have a container full of this optical liquid, and one of the sides of the container is closed with one elastomer membrane, and that's what allows us to change the focal power. To change the shape, what we do is we use these electromagnets to move some of the liquid to the center, and that provokes that the membrane bulges out, or if we remove it from the center, the membrane goes back in. And finally, we need one element, which we call the lens shaper. That's important to get nice shapes of this membrane. We need that the interface with the side of the membrane is really nice and circular, so that we get a nice spherical shapes. With that, we can bulge the lenses out and get focal powers on the positive side, so positive diopter powers. We can leave the membrane in the neutral position and get a flat lens, and we can go on the other side, remove liquid from the center, and get a negative bulge, which gives us the negative focal powers. Also very nicely, when we drive our lenses electrically, due to a combination of properties, the focal power of the lens is linear with the current that we input. So this gives us also a device that is rather simple to drive and then can be combined with other systems very, very easily. And what's more important for our project, I was talking all the time about the speed. I mentioned it very fast, but this is one of the biggest advantage of liquid lenses. For this project, as I was mentioning, we needed to take the biggest sensors possible so that we could take the biggest image and hence less number of images. But that means that we also took the biggest lens we had available in Optotune. That's the one I'm showing in this picture. It has a diameter, the optical aperture has a diameter of 16 millimeters. This is the biggest one. And of course, when we are moving elements, the biggest the lens, the slowest we can move stuff. But even in this situation with the biggest lens, we have response time in the order of tens of milliseconds. If we look at the dark blue line, this is a resonant oscillating system for those that know these topics. So if we try to move it as fast as possible, we can reach our goal focal power very fast. But of course, we'll have a bit of oscillations. That's not the ideal situations. So what we usually do, we use what's called signal conditioning, and hence we get the light blue signal. It's a bit slower, but much more smooth. In this case, what we get is a response time around 5 milliseconds. And if we look when all these oscillations are completely eliminated, that is around 10, 15 milliseconds. Of course, this depends a bit on how big is the step that we are doing. And also, as I said, for smaller lenses, these values are even smaller. But in general, in the order of tens of milliseconds, we can change focal power of these lenses several diopters, and we can do that many times per second. So we could change focal power very fast, take a picture, change again, and repeat that as many times as we need. However, as I said, we have these lenses as off-the-shelf products. So why is Optotune participating as a partner in the research project and not just selling the lenses to the project? And there are two challenges that we have to tackle. The first one is what we call the gravity-induced coma of the liquid lenses. And now I'm going to tell how we solve them. So the liquid lenses are a very simple concept. You have liquid inside this membrane closed compartment. And when they are in what we call the vertical configuration, that is with the optical axis vertical, this is a very passive method. So the liquid is pushed by the motors. That's the active action that we do in our lenses. But then this liquid pushes the membrane. And because the membranes, we fabricate them as homogeneous as possible. And as I said before, the interface is as circular as possible. The membrane deforms naturally in a spherical shape. That is what we want for a lens. However, because we have these moving parts, these passive parts inside the lens, when we tilt the lens, and this is something that happens very often, and in particular in the iToBoS project, you have seen that the arches have different orientations. So the cameras are looking at in different orientations, and hence the lens is also in different orientations. What happens is that the gravity pulls the liquid down, and we cannot avoid that because the liquid is not attached to anything. It's just free inside those containers. And it pushes differently in different regions of the lens. And due to this different hydrostatic pressure, what we get is a shape that is not anymore as sphere as we want, but is what we call a beer belly shape. And as you see, with this shape, you cannot expect a good optical image. In particular, if we go into the optic terms, this shape provokes what is called a coma aberration. And this means that on the top part, we have lower focal power, and in the bottom part, we have a higher focal power. And for most of the time, for Optotune, this has been a limitation of these lenses. We were not able to find a solution to that. And that means that our customers could only use them in vertical optical axis configuration, or if they needed to use in this configuration, they had to accept that the quality was not as good as the traditional glass lenses, which of course have no problem with gravity. However, in the last years, we found a solution for that, and it's actually rather simple to understand. What we do is we add a second container with liquid, and we have a membrane in between the two. So basically, we end up with two liquids and two membranes. If we match properly the stiffness of the membrane and the density of the two liquids and the refractive index of the two liquids, the heavier liquid will push down and the lighter liquid will be pushed up. In the end, because this weighs more, that happens, and the shapes of the two membranes will compensate, and the thickness of the containers that we will get will have exact refractive properties such that at the end, the optical path resembles still the spherical shape that we want. Okay, so basically the two refractive regions will compensate one another, and in the end, we will compensate this coma induced aberration. The activity or the task that we had for iToBoS project was bring that into real lenses. We had proven that in prototypes, in the lab, in small batches, but we had not been able to put that yet in off-the-shelf lenses that we could use in an industrial or in a real life application. And that's what we did during the project. So we went to the EL1640 TC lens to the GTC lens. So the G stands for gravity compensation. And the results are the following. I mean, very fast, very easy to understand. The original lens had this behavior. So when you are at 0 degrees, that's the vertical axis configuration. The wavefront error related to the coma, it's basically negligible. It's only related to the tolerances of the fabrication. But when we were tilting the devices, this was increasing, and in a value that it's not acceptable for the lenses. In the case of the gravity compensator lenses, you can still see, maybe in this graph, it's not perfectly easy to see, there's still a bit remaining of gravity induced coma, but it's very small, and it's in a level where the optical quality of the image is not really affected. So as you can see, this very small data set, but we are able to compensate for that big effect that we had, and now our lenses can be used in any configuration, which is exactly what we needed for the iToBoS project. The other task that we had, I already mentioned very fast at the beginning, is designing this objective module that allows us to take our lenses which can only focus around 0 diopters, so plus some diopters minus some diopters. In the case of the lens that we have been using, plus minus 3 diopters, and translate that tuning to the long distances that we need to be able to image the skin of the patient. But this is actually not that easy, because what we need to know is first, what kind of objective lens do we need? I mean, there are tons of different designs of objective lenses that can focus at long distances, and they have all kinds of different properties. Some of them can focus very far, some others can focus closer, but they can adapt to different distances. Some of them have bigger or higher resolution, some of them have lower. And unfortunately, when we started the project, most of our feedback was qualitative. I mean, our final customer is the physicians, and what they could tell us is, yes, in this image we see these features here, but instead in this other image it's blurry, we don't see the features that we need. So this was a very qualitative approach, and we needed to translate that into numbers to be able to design that objective lens. And what we did is the following. First, we bought some off-the-shelf lenses or objective lenses that exist in the market. Of course, those lenses didn't incorporate the liquid lens, so they have manual tuning, but for this experiment, it was enough. And then what we did is we took images of a real lesion and images of a standardized target. This is targets that are commonly used in optics to see how good is the quality of an imaging system. So we took images of these two elements in different conditions, but always in pairs. So first we took the two images in one set of conditions, then we changed the distance and we took the same images at a different distance. Then we changed the lighting conditions, we changed the opening, the aperture opening of the lens. We used different lenses on the region of where we expected to land. And with that, we were able to translate the qualitative assessment that the doctors were able to give us with these skin lesion images into quantitative assessment using the target images. The result of that, I'm not going to go into details, but was a set of specifications that we could use to design the lens. Of course, working distances, the sensor that was already chosen before, the resolution we want in the target, focal distance length, aperture, MTF parameters, etc. With that, it was time to design the lens. Here, OptoTune, as I said, we don't have the expertise on glass lenses. We do the liquid lens. So usually in these cases, we partnered with some companies that have that expertise. So in this case, we went to one of our partners, and with the specifications that we had found, we could together design this objective. Once it's fabricated, it looks like this. And again, I'm not going to go into details due to the time, but you can see here, for example, the modulation transfer function. You can see it's above the values that I was saying in these specifications here. So With that, we have a lens that, first of all, can focus at long distances with this nice objective lens. And at the same time, it incorporates in this region of the lens our liquid lens that allows this fast focusing, and hence we can try to scan as fast as possible all those number of images that we need for the iToBoS scanner. With that, I arrived to the end of my talk. So now I want to thank you for your attention, and I'm open to any question that you might have.
Great, thank you. That was really cool to see how things have evolved. And it is like all the questions we had been wondering about the liquid in the lenses, that was exactly on. So I've seen a few people have already added comments to the chat. Do you want to bring this up now and ask directly?
Maybe like, let me open it. I'll start from the top.
I thought that the people could also just say what they wanted to say.
Well, I'll start then with this thing, because I think Violetta was also asking similar things. So I think it's really cool how you've managed to sort out this problem with the two different liquids. So I think that's really cool. But then we're also talking about something that's moving. So it has to basically, so you've got great reaction times, but is that also going to be compensated by the fact that these things are not just vertical, they're sometimes upside down, sometimes this way up? Is that going to work fast enough with this movement of the two membranes in sequence?
Yes, exactly. So I'm going here. Go to the here. So yes, I mean, this is the timing for the original lenses. And you are right to point that when we put a second liquid there, it becomes a bit slower. That's clear. However, in the iToBoS project here, we have an advantage. And it's the fact that in most of the situations, or most of the movement that we'll have always very small steps. So first of all, the camera is in a linear stage, and it's moving along this linear stage, but it only moves little steps, because every time we want to image the part of the body that is next to the last image we took. So there we do a very small step, and that means that the liquid also feels small accelerations from that small step. Even if it's a fast movement, the total force felt by the liquid is not too much. And secondly, about the orientations. This liquid allows us to change the orientation, and after a few milliseconds, all the liquids are again equilibrated. So you see there will be some oscillations. That's clear because it's a dynamic, springy system. But after a few milliseconds, that's already stabilized. And in the iToBoS case, there is another advantage, and is the fact that the orientation will not change actually. So we need the gravity compensation because we are not in a vertical configuration. We are at some angle looking at the patient, but the movement will be linear, and that orientation doesn't change too much. So it's necessary to have it, but the speed of the compensation is not a limitation for that project. It's the speed of the overall lens that we need to have, so that there's little steps that we are doing are still possible. But the gravity compensation is not making the lens too slow for this application.
Also related to that, yeah, thank you, Gili, for addressing my question too. Related to that, I was just to align my thoughts in this technical field. So you said about focal power, and this impressed me very much, because I remember as a microbiologist, I was very bothered when I was changing for the microscope, the objectives. And then I was just staying there waiting to see clear what I wanted to see. So, and this impressed me that you still preserve the focal power even if you move. And I was wondering, is this focal power and the speed you regain the focal power influenced by this horizontal position? Because I didn't get this from the.
Okay, well, I was moving here because I thought it would be more on this side, but well, I can answer anyway. Yeah, so the quality of the image, it doesn't matter in which position we are. The quality is always the same. I mean, there is some variations, but they are all, so the specs that we give on the optical quality are the worst case possible. So in the extremes, when the membrane is really blown up, the quality is slightly worse than when it's flat. But all of them are better than the base specification that we give in our specification sheets of the lens. So when I was showing, I'll go back to here. When I'm showing this is already on the worst case possible. So
okay,
at this point, when we put the specification, for example, in the original lens, we would say that the lens has a wavefront error below 0.2 for the vertical orientation, no matter what is the optical power. That means that maybe at 0 we are here, and then we go a bit high when we have a bit of optical power, but always below that value. And the problem with the horizontal was that we were going much higher than that, like double or triple that error. And now with this compensation, again, we'll have variations depending on exactly the optical power we are using, but they will be all below the same thresholds we had for the original lenses.
And something maybe silly, how many cameras will be actually into the iToBoS scanner?
So this is a tricky question because I mentioned it very fast because I didn't want to enter in that topic, but due to the delays in all the projects we have having, the original design that I showed at the beginning, that was these 5 arches with 3 cameras per arch, so a total of 15, might be changing. I know that for a first prototype, Bosch will be using robotic arms instead of this arch architecture. But I have to admit, I don't know if that will be the final design or it's only such that we can get images already and start working in other packages.
We are very curious about.
Yes, I think when you have the webinar on the architecture of the whole scanner, they will be able to tell more. But the initial idea was 15. In the end, we want to reduce the time. So a part of scanning very fast and moving these cameras very fast, another solution is just having more cameras such that each one is responsible only for a small part of the body of the patient. So that was the idea.
Any other questions for Xavi?
Let's see what was in the chat. About the space. Again, this depends a bit on, maybe Bosch can answer much better that. This will not be a small scanner. In the end, a full patient has to fit inside the scanner. So I was putting an example, an MRI. Usually those are even bulkier because there is a big magnet, but still it will be a device that will be able to cover the full body of a patient. The final size it will take still some years to define. Now we are going into prototypes. When this device goes into production, for sure in between we'll have had a couple of iterations. But in the end, if I go back to the initial pictures I showed, it was shown the first designs of the scanner. Let's see. Yeah. So this is a very initial prototyping or designing sketch, but you can see already that if a human being has to fit there, it will not be a small one. Still, it will be a big device. The goal is that at least it's an all-in-one device, that with only this one, we got already enough information such that the clinicians have all they need to take their decisions. So that's the important one. How you keep the liquid doing what it should when cameras are swiveling and moving. Yeah, this is an interesting one. So our cameras, and especially in this application of iToBoS, we are not taking video, we are taking pictures, still pictures. So while the lens is moving, while it's feeling the acceleration of a changing movement, it's clear that our lenses are not in a stable position. The important thing is that once that acceleration disappears, and I'm talking acceleration, not velocity, so it could be in a movement, but it has to be a stable movement. When the acceleration disappears, then the liquid stabilizes in matters of milliseconds, tens of milliseconds. So that's what we use in this case. If you would have these cameras or these lenses in a device that is moving all the time and changing directions all the time, that means having accelerations in different directions all the time, you would always need to have a moment where your movement is stable, be it completely stopped or be it stable, constant velocity, such that the liquid can stabilize and then take the pictures. Okay, so this is really clear for our lenses. They need a bit of stabilization, but again, it's on the order of milliseconds. So usually it's not a problem for most of the systems, and in particular for the iToBoS. Let's go to the next
Martina
one.
had a question, I think. Martina.
Down here. Influence of prismatic. Yeah, I mean, as any lens, prism is something that appears usually through the tolerance of the assembly of the different elements. So in most in classical lenses, you have different glass elements, and if one is tilted with respect to the other, you start to get prism and other kinds of aberrations. In our case, we do have a glass in one of the sides. So the container is usually a metallic or plastic container with a glass on one side and the membrane on the other. So yes, these two surfaces need to be as parallel as possible. This is important. And then when we put our liquid lens on the big glass module, also we have the same conditions. We need that our liquid lens is parallel with respect to the other glass components. But this is something that in optics is always a topic. So our lenses don't introduce more of these challenges. They are just another element that we add in the big glass module. Yeah, prism is not extra in this case.
Thank you.
Went through some.
I have another question actually. So first, thanks so much. So I've been wearing glasses for most of my life, so I'm kind of partial to optics in a way. I've also been working with them, so I've always been fascinated like with microscopy. But something that kind of stuck me when we started this project is like, I have worked in specimens. So usually what you tried is you try to keep your camera as stable as possible, and you would ideally move your object. Now, of course, you can't turn a patient around under your fixed setup, right? So I thought that was like, that I thought was kind of, I remember thinking about this, how the approach or how it changes your thinking when you go into human. Now, you said this earlier on that, if it was just a simple product, you would have just sold the lenses to the project. So this was something novel. I'm very interested in, like, you are in a company setting. So how do you integrate this with the research link up like here? And what do you think, what was the most, exciting part for you of being part in this?
I mean, it's clear that as a company, we always have our plans, and this has been actually a topic that has been tested in the lab for already a couple of years. But you can understand that these lenses, I didn't talk about that, but the biggest challenge that we have in Optotune is process. We have several materials interacting. These liquids are very special liquids, and also the membranes are special materials, and then they have to be attached together. So there is a lot of chemical interaction. It's not my topic, but basically a part of the physics that we have to design the lens with the right parameters. Building them is really a challenge. And what was stopping us from having this solution that we had proven theoretically and in small prototypes, to bring that to a real product, the effort of investing in all this process was missing. And I think here is where the iToBoS project arrived in the right moment for us. The iToBoS needed that solution. We knew how to solve it, and we just needed time. And yeah, in these projects, there's always the money factor. So it was a perfect match of the project needing this solution and us just needing the time and resources to invest on that development. So basically, it has been the opportunity to do all these ideas that we have in a high-tech company, as it is Optotune, that you put in a drawer because at some point you have to choose which ones you do at every moment, and you cannot invest in all of them. And that's something very hard to do because some of them are really cool ideas, but they are difficult, they need time. And this project has given us this opportunity, basically.
Now, Bettina inspired me with another question for you. I remember our talk during the last meeting, and one of the subjects was how, with this technology that you are bringing in iToBoS, do you think that we will be able to avoid thermoscopy I mean, the ideal of iToBoS at the end to not having dermoscopy, so the image quality and accuracy will be the same or higher at the end.
Exactly. So that's the whole goal. So when I was mentioning before that the clinicians were being the referees to say if an image was good or bad, basically they were trying to say, is this image comparable to what we get from other dermoscope, or it's not? Basically, that's the final goal. What we want to do is that this avatar, this digital avatar of the body of the patient that we will be at the end, that we can zoom into it the same way we are used to do in Google Maps, that you see first the big picture, and then when you find the region you are interested on, you can zoom and get that part of the image expanded and still see all the detail. So the idea is that the final tool for the clinician is something similar to that, an avatar of the patient where you can have different lesions tagged. And as a clinician, you can just zoom in that area and see the details and find the high quality images taken by the iToBoS and see all the detail on them without having to take local images with a dermatoscope. And then, I don't know, in your file system, link the big image from the full body scanner with the small image of the or local image of the dermatoscope. That would be the final goal. We believe that we can do that. Yeah.
Yeah, so that would be actually quite useful then, because you could link then, or you could flag in a way the things that are even difficult for the dermatologist with a dermatoscope to identify, like your amelanotic melanomas, which need different lighting, I imagine, to really pick up the features, and difficult spitzoids and things like that. Now, it's not really fair to ask you as not as a dermatologist, but I think just being able to have that avatar where you have some question marks, like, you know, this may be a lesion, it needs to be looked at specifically. I think that would be really cool.
Yeah, and also on the lines of what you are saying, again, not exactly my topic, but the idea also with the artificial intelligence assistant is that you take a full scanner one day, and then the next month you have another visit, you take another scanner, and the system is able to go to those regions that had them all, but you were not sure if it was a lesion or not, and see exactly what is the evolution. And doing this automatized makes that the doctor doesn't need to go through all the body, because the machine can already detect, okay, here there is a spot, this needs to be controlled. And this is something that we expect to be able to do with this system. Okay, I think more questions appear at the end.
I just added that, please, everyone, if you're interested in or would like to see more or hear more about it, please, there is a link to a feedback form. It's just like for us to collect what else would be worth kind of going a little bit deeper into. So I personally would just like love to see these lenses, because it's just like, you know, just like it's like a drawing is one thing, but to see what they look like in real life, I think is fascinating, and to see what you get out of it. I also think that thanks for that answer, because in a way I think you probably in a way were like a solution in want of a problem, right? So you had this thought, you thought, okay, this is how we could solve it, but if you don't have a concrete problem to go after, you wouldn't be doing it. So I think there is something for us as patient advocates, as learnings in this in this project, apart from it being cool technology, is like if we think about Rafa's talk at MPNE 2023 on how he got into the topic, and that we started from monitoring underwater ground to monitoring lesions on a body. I think there's also something in there. So that technology that maybe that wasn't planned for this ends up making a real difference. So I think there is something in this leap or this application that I think we haven't talked much about in our community about. So it's something we definitely should have on our, I don't know, I can't phrase it more concretely, but in terms of, you know, finding technical solutions to the issues we're having that could move the field forward.
And I also like this idea of avoiding the black box, because I like what you said about the fact that explainable, I can give an explanation actually for the findings, because what is upsetting me sometimes is just receiving outcome without argumentation. And I think this is helping patients and patient advocates to stick with the solution, or even treatment or intervention, healthcare intervention. Without an explanation, everything is like, why I should do that. So I like this avoiding black box that,
Yeah,
the project brings up.
Yeah, again, it's not my topic, but I agree
Yes, I
completely.
know.
I mean, doctors have to be anyway the last person taking the decision. So this should be only a guide for them or a support for them. So if it's just a percentage, I don't know, if it's a 60% percentage, what does it mean? It's good or bad? Because it's very close to 50. So having some more information behind that, saying yes, the system has decided that because this region of the mole looks a bit like these other examples that you gave me as a training. That's what the doctor needs, a bit of explaining why that value has arisen. So yeah, I think it's really cool. I actually had no idea this existed or this was a field of research, and I'm also really interested.
Yeah, but think about this application, very common application that are on the market, like skin vision. I mean, you put your images there, and it's coming back a decision, and you are looking, should I trust that? Who took that decision? And it's like, why you did it like this? Why you how you arrive ended up? Okay, you can think about ABCDA criteria or something else, but could be more things that was in that image leading to the decision that, okay, your mole is not harmful. So all these things are just fascinating. Of course, it's not the subject of today, but it's just something to think about.
And if you think of the handhelds,
perfect.
yeah, sorry, the handhelds also. I mean, you think of the errors that come just because people have dirty lenses or anything like that. So if you're relying on those kind of ones, I think it can be quite tricky.
Absolutely.
So actually, that's the perfect advertisement for one of the next sessions we have planned, namely on explainable AI, that will hopefully help us to deal with some of these issues. I hope, Xavi, that your lenses will not be dirty, because I think that's an issue that we have discussed not in these handheld devices that we all have. But maybe that could also be something interesting to discuss in the future. So if we think about these data sets that we will generate in a way they're probably now ideal or the highest quality we can get, but what happens if we go into a different type of market? So I think with that, there's a lot of food for thought. This is the perfect moment for any last words, last questions from our participants. Thank you, everyone, for coming, for making time. And as previously said, we will make the recording available on the iToBoS YouTube channel, and we will also alert everyone once they're up there, so you can find them and share them with your networks. Thank you, and bye-bye.
Thank you, everybody.
Thank you.